Hello students, welcome in this today's session. Today we are going to see your class 12th full syllabus. What is you are going to learn in class 12th, right? So as you know that class 11 and class 12 is a combined +2 syllabus. So 5 units you have learned in class 11 and next 5 units will be there in class 12th. So 1 to 5 units you have completed in class 11. Now 6, 7, 8, 9, 10. These next 5 units you are going to study in class 12. So let's see the class 12th syllabus of year 2020-21. Let's see your class 12th syllabus. You have in class 12 next 5 units. Unit 6, 7, 8, 9, 10. Unit 6 is reproduction, carries total 14 marks. In that 4 chapters are there and these all 4 chapters together carries 14 marks. Not an every single chapter's weightage is given. Next unit is of genetics and evolution. It has 3 chapters and all the 3 chapters together carries 18 marks weightage. Unit 8 is there biology and human welfare. It carries again 14 marks. Unit 9 is there biotechnology and its application, which has only 2 chapters and carries 10 marks. Chapter wise if you will see, this unit will have the highest weightage of each chapter, that is a 5 marks. But we don't have the chapter wise weightage, unit wise weightage. Last unit is there, that is ecology and environment. It carries again 14 marks. So this is your 70 marks distribution among these 5 units. 6th unit 14 marks, 7th unit 18 marks, 8th unit 14 marks, 9th unit 10 marks and 10th unit is of 14 marks. So total this is of a 17 marks distribution. Now just see the unit 6 reproduction. As I said earlier, this unit has 4 chapters. First is reproduction in organism. In this general reproduction in the organism you are studying, maybe that is in the microorganisms like amoeba, yeast, then in some of the animals, in plants, vegetative reproduction. So general way of reproduction you are going to learn in this chapter. Second chapter is that sexual reproduction in flowering plants. Now here in this chapter you are going to learn only the plant's reproduction, how it takes place from the reproductive part in the plant, that is flower, to the fruit formation. Next is human reproduction. In detail you are going to study in chapter 3 and chapter 4 is of reproductive health. So this all 4 chapters gives your first unit, and for this you must have studied the base related to this unit in class 10th. So whatever you have studied in class 10th, next part of that chapter is given in the 4 chapters in class 12th. Next is unit 7 genetics and evolution. In that you are going to study the 3 chapters. Chapter 5th is the principles of inheritance and variation. And again this is the base of this chapter you have studied in class 10, that is Mendelian, monohybrid cross, dihybrid cross, variations and inheritance. This all you are going to study in this same chapter, and something new advanced, little bit ahead part is there, that is in detail you will study in this 5th chapter. Next is that molecular basis of inheritance. This chapter is related with the DNA and RNA structure, the gene expression, human genome project and DNA fingerprinting. So more new points you will get to learn in this chapter, and this is again very important chapter from the point of exam. Next chapter from this unit is the evolution. By the evolution term you can understand here that you are going to study the evolution of earth, evolution of life on the earth and evolution of different types of animals on the earth. At last you are going to study a short human evolution. So this is all related about the evolution you are going to study in the evolution chapter. Next unit is there, the unit biology in human welfare. So in this you will come to know here how the biology is useful for human welfare. So for that you are going to study the chapter 8, human health and diseases for the cell unit of life. In that you are going to study the structure of a cell. So this you have studied in class 9th. That was the base, the cell which you have studied, the different cell organelles. The same you are going to study here as an introductory and revisional part, and little bit more detail up to the class 11th you are going to study in chapter 8. Chapter 9 is there, that is of strategies for enhancement in the food production. So in that again the base is there in the class 9th, that is plant breeding and animal breeding. The same you are going to study in this with little bit additional and advanced part. Last is that microbes in human welfare. Now by the name you can understand that we are going to study the different types of microorganisms and how they are useful to us and how we are using them in various fields. Now the base for this chapter is there in class 8th, where you have studied the chapter microorganism, friend and foe. There also you have studied how the microorganisms are useful for us. The same you are going to study but little bit at higher level and little bit advanced in class 12 in chapter 10. Now see the next unit, unit 9, biotechnology and its application. Now this is the new portion for you. This is the new topic for you. You don't have studied any BS related to this. So this is a very interesting topic, and in this only 2 chapters are there. Biotechnology, its principles and processes. So what is a biotechnology and how it takes place? That whole process you are going to study in this chapter 11, and the next chapter is fully based on its application, where this biotechnology we are going to apply in the various fields for our welfare, for human welfare. So that we are going to study in chapter 12. So only 2 chapters are there in the unit 9. Very important unit 9 from the exam point of view. And your last unit is there, that is unit 10, ecology and environments. Very simple, very easy and based on the general science. You have studied the general science in class 9th, class 10th. Now also you are going to study in general science in the 4 parts, 4 chapters. Chapter number 13, organism and the population. Here you are going to learn the organism and their populations and their interaction. Ecosystem you are going to learn here, right? How they are interact with each other positively, negatively, mutually. So this is a very interesting chapter and very easy. Next is that ecosystem chapter is there. So in this you are going to study the different types of ecosystem and the various changes takes place in the ecosystem. Energy flow is there, that you are going to study. Pyramids are there, that you are going to study. So again it is very easy and very interesting chapter, ecosystem. Next is that the biodiversity and its conservation. So this is again very important chapter, very an interesting chapter, where you are going to study about the biodiversity. Because of that what our universe is stable and we are living from such a long time on the earth, just because of we have a diversity, biodiversity, because of that. So what is mean by biodiversity, why it is important, how it is conserved, why we need to conserve, how it is preserved, that all things you are going to study in chapter number 15. And our last chapter is environmental issues. So this is again very easy chapter, that is here in this chapter you are going to study the water pollution, air pollution, land pollution, radioactive pollution, noise pollution, some of the case studies related to that, and it's very easy. So overall we can say that our 12th standard syllabus is very easy and very short. Many of the chapters base already you have studied in 9th and 10th, and little bit more you are going to study in class 11th. If you compare your syllabus of 11th, then again you will see that it is very short as compared to the 11th. So it is very yes. Now based on this syllabus you are going to give exam. Now let's see about the exam. Based on this all 5 units you are going to give 70 marks written exam. Now let's see your paper style, how it is going to be there in the board. For this year CBSE has made some changes. So in that they have given total 27 questions for 70 marks, and remaining 30 marks are there that will be taken for the practical exam. Now see, in the 70 marks there are total 27 questions, and all these 27 questions will be asked in 5 sections. Now in this no overall choice is given. There is an internal choice and all questions are compulsory. Now if you will see the internal choices, then 2 internal choices will be there in 1 mark question, 2 internal choice will be there in 2 marks question, 2 internal choice will be there in 3 marks questions, and 5 marks 3 questions are there. So for that all 3 questions internal choice will be there. So let's see the what is asked in 5 sections. So the section A is there. In that 5 questions will be asked, that will be of MCQ. This year CBSE has said that 20 marks objective questions will be asked in most of the all subjects, but not in biology. Biology only 5 MCQ questions will be asked in section A. So your section A is of MCQ. Now the section B is there, that contains 2 marks questions, and that will be asked 6 questions. So your section B will have the 6 questions of 2 marks. In that internal choice will be there. Now section C is there. In that 8 questions will be asked. Give the answers in short for 3 marks, right? And in that also you will get 2 internal choices. Your section D is there. In that 3 marks question, 3 questions will be asked, and these 3 questions will be asked based on case study. Now this case study based questions means which type? Generally these questions will be based on the diagrams which is there in your textbook. Based on that diagram the questions will be asked. And then last year section E is there. In that 5 marks questions will be there. So 3 questions of 5 marks will be asked in section E, which will give you the descriptive answer or long answers, or sub questions are there where you have to answer in short. So in that way total 27 questions will be asked in this 5 sections. So this is your paper style. Now see your remaining 30 marks that will be conducted in a practical exam. Now for the practical exam external is going to become, and your how the practical exam is conducted, that see the first. So before that see your syllabus for the practical exam. So for your practical your all experiments are divided into 2 parts, part A and part B. The part A is based on the experiment which you have to perform. So in that total 9 experiments are there. So your first experiment is that prepare a temporary mount of pollen germination. Very easy, and you know that here slide preparation is there. Remember this whatever I am saying, at last you will come to know. So your first experiment is of slide preparation, and you have to prepare a slide of a pollen grain. Your next experiment is based on the soil. So you have to collect the soil sample from at least 2 places, and you have to study both the soil samples for the texture, moisture content in the soil, pH of the soil and water holding. So 4 sub points are there in this practical. So these all 4 sub practicals will be considered as your second practical based on soil sample. Your next practical is that the water practical. So this practical is based on the water sample. So in this practical you are studying the water sample which is collected from the 2 different water bodies around you, and these samples you are going to study for the pH, then clarity and the presence of living organisms in them, right? So 3 points you are going to study for the water sample: pH, clarity and presence of living organisms. So this is your third practical. Next practical is that the study of the presence of suspended particulate matter in the air at 2 widely different sites. Means 2 locations you have to select, and what is the percentage of the dust particles, suspended particles in the air, that you have to study. Now your fifth practical is study of plant population density by quadrate method. Now in this you are going to select one quadrate, and in that different plant species, what is the density is there, that you are going to calculate. Your experiment number 6 is there. In that you are going to study the plant population frequency in a quadrate. So same as like earlier, different species you have to select, count their number and calculate their frequency in each quadrate. Very simple experiment. Next is that experiment number 7, that is prepare a temporary mount of onion root tips to study meiosis. Once again here you are going to study a slide. You have to prepare a slide, right? So in this practical you have to prepare a slide of onion root tips. So in this you are going to study the different phases of cell division, that is prophase, metaphase, anaphase and telophase. So here you have to prepare a slide and show the chromosomal arrangement in 4 different phases, right? So this is one more slide preparation practical. Your next practical is practical number 8, that you are going to study the effect of temperature and effect of different pH on the action of salivary amylase on starch. You know that starch is a carbohydrate and this starch is digested by salivary amylase enzyme. So whatever its activity is there of digesting starch by the salivary amylase, how the temperature will be affect on this process and how the pH will affect on this process, you are going to study in your experiment number 8. Next your experiment is the isolate DNA from available plant material. So some of the examples are given here, that is spinach, green pea seeds or papaya. So any plant material which is available to you, you can take that sample and procedure is given. According to that procedure you have to extract the DNA of that plant material. Very simple, very easy and very interesting practical. So these are all 9 experiments of your part A, and as I said earlier, your part A of the experiment where you are doing the experiment, so that you have to do, which will be asked in exam for major practical and minor practical. Now we are going to see the part B. See, your part B experiments are there. These all experiments are the spotting experiments. Means what? Here you have to just observe the specimen and you have to comment on that, write something, draw the picture. Means you have to just spot that specimen. Here you don't have to do any experiment observe and record. Nothing is like that, right? Whatever available material is there, that you have to just observe. And based on this full part B experiments, 7 marks spotting question will be asked in a practical exam. So this portion B is there of experiment is carries total 7 marks. Now in this see your experiment number 1 is there, that in that you are going to study the flowers adaptation. Now the flowers are adapted for pollination according to their agents. So there are various agents for the pollination like wind, water and insect. Now here you are going to study their adaptations, how they get adapted for pollination according to their agencies. So you are going to study here wind pollination adaptation, insect pollination adaptation and bird pollination adaptation. Your next experiment is pollen germination on a stigma through permanent slide. Just remember, your first experiment is there in part A. There you are again studying the pollen germination, but there you are preparing your own slide, and here the slide will be permanent and just you have to observe it under the microscope and you have to study, right? So this is what the difference. So this is your second practical from part B. Your next experiment in part B, that is experiment number 3, it is just based on the tears of the ovary and the tears of the testes. These permanent slides you have to observe under the microscope carefully and study them, right? So your third experiment is identification of the stages of gamete developments in the testes as well as in the ovaries. Your experiment number 4 is of meiosis in onion bud cell or grasshopper testes through permanent slides. Now you again remember that mitosis we have studied in part A. There you have to prepare your own slide and the plant material was the root tip of onion. But here you are studying the meiosis, that is reductional division. And you know that the reductional division takes place only in the gametic cells. So the gametic cells means where the gametes are formed, maybe in the anthers or in the ovule. So here you are going to study the onion bud cells, means anther section you are studying, and how the pollen grains are formed, haploid pollen grains are formed, their reductional division takes place, that you are studying with the help of plant material, that onion bud cells. Or if you want to take an animal cell specimen, then here suggested specimen is grasshopper testes. Here also this is the gamete cell where the male gamete is forming by reductional division. So these are the two specimens. Any one of them you can use to study the meiosis. Your next experiment is experiment number 5, that is tears of blastula through permanent slides. Here you have to study the blastula. When the embryo is developing, so in that developing embryo, blastula is one of the stage. So that stage you have to study through a permanent slide. So any mammalian's permanent slide will be available in the lab, that you have to observe and study. Your next experiment is, your experiment number 6 is Mendelian Inheritance using seeds of different color or size of any plant. So generally here we are using the pea seeds. So what you have to study generally are the Mendelian inheritance. Mendelian monohybrid cross and dihybrid cross, they said that the parental characters inherit into the next generation. Whenever there is a homozygous dominant male parent crossed with the homozygous recessive parent, then their first generation is there, it will be showing completely dominant characters, and their F1 generations, whenever it is self-crossed, it will give 3:1 ratio. So that 3:1 ratio is there. Sometimes it is 1:1 ratio is there, or sometimes all shows the dominant characters. So based on that, we can try to identify what is the genotype of their parent. So here either using the color of the seeds or size of the seeds, means round and wrinkled or yellow and green color seeds. We are studying Mendelian inheritance. Your next experiment is experiment number 7. Here you are studying a pedigree chart of any one of the genetic traits. So genetic traits are given here: rolling of tongue, blood groups, earlobes attached or free, and widow's peak. So out of that, any one genetic trait you can consider, and for that trait you are going to study a pedigree chart which will be provided. Here color blindness is also there. Next your experiment is there, experiment number 8, that is controlled pollination, emasculation, tagging and bagging. So in that, generally this is of hybridization state, and where the 3 steps are there, that is emasculation, tagging and bagging. So how it will be carried out, when it will be carried out, so that you are going to study in this experiment. Out of that, any one stage will be kept in the exam. Experiment number 9, that is common disease causing organisms like Ascaris, Enchamoeba, Plasmodium, any fungal disease like ringworm is there, and all these diseases you are going to study with the help of permanent slides which will be available in the lab. So in this way, based on the disease causing organism, this experiment is there. Next experiment is there, you have to study the 2 plants and 2 examples of animals from the xeric condition. Now here the lab preserved specimens are there, that is totally banned by the government, even the CBSE. So here you can found their models or the pictures, and with the help of them you are studying the xeric conditions, modifications in their morphological adaptations of the 2 plants and 2 animals. Same here, your last practical is there, practical number 11, that you are again studying the 2 examples of the plants and 2 examples of the animals of aquatic condition, found in aquatic conditions. As they are living in water, what are the adaptations are there to survive betterly in the water? That adaptations you are studying here. Again here you have to use a model or a picture, not a specimen. Now these are what your 11 experiments. Here based on these 11 experiments, 7 spot from the part B will be asked for 7 marks. So from the 11, any randomly 7 experiments will be chosen, and these 7 experiments, one specimen will be kept for a spotting. Now see practical pattern. What are the questions and which type of practical questions will be asked in the exam for the practical? So here practical pattern is, as you know, practical carries 30 marks. So in that, first question is of a major experiment. It will be asked, and that will be asked from your part A. And here also for major, even the number of experiments are given, that which number of experiments will be considered to ask in this 5 marks question. That is experiment number 5, 6, 8 and 9. Only these 4 experiments are there. From that 4, any one will be asked for major experiment for 5 marks. Now next is that minor experiment, that is also from the part A, and it will carry 4 marks. Now for this 4 marks experiments, again it is given second experiment, third experiment and fourth experiment. So out of these three, any one will be asked for 4 marks. The third question is slide preparation. This is again asked from the part A, and you know that I said during that experiment here you have to prepare a slide. So in our syllabus only first and seventh experiment is there where you have to prepare a slide. So the first experiment was pollen germination slide and the seventh was the meiosis slide. So any one will be asked in the slide preparation, that carries 5 marks. Slide preparation will be carries 5 marks. The next is that spotting. This question number 4 is fully based on part B, and these 3 questions are there, that is from the part A. So this is the spotting, carries 7 marks. Next, practical record book is there, means your journal, whatever you have written the experiments, your observations, diagram you have drawn, that practical record book whatever you have mentioned throughout the year, that carries the 4 marks. Based on these all practicals and even whatever you have mentioned in the record book, based on that viva will be conducted. That viva plus practical record gives the 4 marks. And last, project record. You have to do one investigatory project. So that investigatory project record and viva based on this project together carries 5 marks. So in this way total 30 marks practical will be conducted. Now we have seen our full syllabus for year 2020-21. If you compare this syllabus with the earlier year, then there is not a major change. Only for this year the term scared grew is added in chapter number 15 and there is no change in the practical. Now as you know, the COVID-19 disease is there. The whole world is suffering from this disease, and as lockdown is there from last 2-3 months, so our schools are closed, and that's why online teaching is going on. Whenever the schools is opened, that time our syllabus maybe we have to rush up fast. So government has decided to cut off the 30% syllabus. So whenever it will be updated, I will be there with the new updates to cut off that 30% syllabus. Till then you follow this syllabus and do well. All the best.